Hello everyone. Welcome to the fourth talk of the function. Today we will be talking about cellular assays, dynamics, and ligand virtual screening towards understanding protein function. Over to you, Meenakshi.
Thank you, Shailya. So welcome all of you and to the 13th talk in the CAPS talk series. So we are in the fourth talk in the function theme. I'll share my screen. Yeah. So in the last week, we had discussed about, in the function theme, we had discussed about churning algorithms to mine protein functions. And we had discussed several algorithms, such as using conserved functional motifs, then using chemical residue conservations. We had also looked at a combination of these methods. So we had also looked at a combination of these methods, such as using functional motifs and phylogeny for functional annotation. So today in this theme, we move on to the next topic, which is cellular assays, dynamics, and ligand virtual screening towards understanding protein function. So for today's theme, we'll be discussing cellular assays, using cellular assays, molecular dynamic studies, and virtual screenings to understand protein function. So we have several proteins today being discussed. So we'll start with Sauradeep's so he was a PhD lab and the Dasaradhi lab in InStem. So Sauradeep has worked on the DDX24 protein, which is a dead box RNA helicase protein. He has looked at the function of this protein in planarian regeneration. So he will talk us about his findings and the cellular assays that he has used for the same. And please stay tuned. Very cool staining images and microscopy images from Sauradeep, and to hear the cool stories. Over to you, Sauradeep.
Thank you, Meenakshi. I'm so happy to be back talking with you all after exactly 1 year. And even though this is like really late at night, but I'm really hoping like this would be fun. Okay. Good afternoon, everyone. I'm Sauradeep. I'm an ex-student of our Lab 25, and I'm going to talk about one of the major projects that I've done during my PhD. Now, planarians are a great model system to study whole body regeneration. Any part of their body which is lost to amputation or injury is fully and functionally recovered in few weeks, which is a marvelous thing, right? Now, at the heart of such a dramatic event lies a series of phenomena which is outstanding, which is their ability to re-establish polarity as well as positional information re-specification. To show examples, here you can see SFRP1, which marks the head tip. And you can see very early on in regeneration, as early on as 3 hours post-amputation, that SFRP1 positive cells are being marked, and this region would become the new head. Similarly, a posterior morphogen, WNT2, has a tail to trunk expression domain. And during regeneration, you can see progressively the expression domain gets rewired, and in the newly formed tail, the allometry is properly established. In a nutshell, these different phenomena, positional markers and morphogens and polarity molecules, they establish a three-dimensional coordinate system that tells the regenerated tissues where to go and make the appropriate structures. And what happens in absence of these processes is that you have these stunning homeotic type transformations, like for instance, a posterior pharynx popping up, or in place of a tail regeneration, you are having a head regeneration, or two tails regenerating in case of APC1 RNAi. So clearly, you need bona fide positional information re-specification for proper regeneration outcome. Now, when we are talking about these morphogens, it's almost impossible to talk about them without mentioning one specific tissue type, which are the muscles, the body-wide muscles. And body-wide muscles in planarians or all these flatworms exclusively express all the positional determinants, fold determinants, these morphogens. Now, if this sounds peculiar to you from a developmental biology standpoint, it is peculiar. This is a very novel function which is associated with muscle. But this is not where the peculiarity of these flatworm muscle ends. The flatworm muscles also function like connective tissues. They secrete almost everything associated with extracellular matrix composition. At the same time, they form structural scaffolds associated with organ regeneration. And of course, they have a massive role in locomotion. Therefore, given their profound versatility, a lot of studies have relied on understanding the cell biology of these muscles as a toolkit to understand regeneration per se. Now, it was around this time, our lab, back at our lab, we were performing an RNAi screen for dead box helicases. And these translation regulators, these dead box family of proteins, are a critical function robustly to regulate the translational output and therefore regulate muscle mass and function. And all of these are muscle expressed. So during our RNAi screen, the first candidate, DDX24, the one that showed us the major defect, eventually became the nucleus of our story, and which is what we're going to present here as well. Okay, so we raised a custom antibody to see if indeed DDX24 that we found from our transcriptome resources was muscle expressed. And what we found was that indeed DDX24 had a strong expression in muscle with preferable enrichment in longitudinal and diagonal muscle fibers. And we corroborated this with our double RNA fish that indeed DDX24 cells were highly positive for muscle markers. And we have used multiple muscle markers to validate this. One shown here, this is PWM is the second one, and MyOD being the third, that indeed that is the case. Now, if indeed this is muscle expressed, and we know muscles have a big role in the morphogen gradient rescaling during regeneration, then the next natural question to ask was, what happens to regeneration if you knock down this gene? And no surprises, regeneration was actually messed up when DDX24 was knocked down. And we validated the messed upness of this regeneration defect by staining for eye markers, here shown by Ariston and Opsin, showing either it was rudimentary eye regeneration or you had like almost no eyes. Gut regeneration was similarly impaired, here shown by the black arrows. That the new gut branch is missing from these animals. Padding's regeneration was impaired. It's the organs through which they eat as well as they throughout processed food. At the same time, you had rudimentary brain regeneration. So yeah, all in all, DDX24 animals had severely compromised regeneration. Now, DDX24 is expressed in muscles, right? So the next natural question for us to ask was, what is happening to muscles? And if you take a later time point of regeneration, the 7-day post-amputation time point, we saw that muscle fiber organization was really compromised. And this is not, by the way, a hole in the animal, as you can see that the hook staining looks continuous. It's a specific muscle fiber architecture defect at 7 days post-amputation. Now, 7 days post-amputation is a pretty late time point, right? A lot can happen before that. So we wanted to follow how exactly does this phenotype manifest. So we did a time course staining of this, and we got two phenotypes. The first one is that till 3 days post-amputation or 72 HPA, the defect in muscle fiber organization was predominantly located at the tip of the head. Whereas at later time points, it's as if like the organization defect comes inwardly, and we see a similar defect as we saw at 7 DPA. So it starts at the tip, and it's sort of like it sort of ingresses and by 7 days post amputation. Now, the first part of this defective trajectory was something that really caught our attention. And it caught our attention because at the blastemal surface, or at the site of regeneration, I mean, till 72 hours post amputation, there is a respecification of a very specific kind of cells called the anterior pole cells, here shown in green, and it is sort of tethered in the entire muscle mesh shown in magenta. Now, these pole cells are the organizer equivalents in these flatworms, meaning that the specification of these pole is absolutely necessary for organ regeneration, but even more so for organ patterning and positioning and allometry establishment. So now that till 72 HPA or 3 days post amputation, the muscle fiber at the tip is damaged, we wondered, are the anterior pole cells being specified properly? And not surprisingly, we see that no, in DDX24 knockdown animals, anterior pole cells, as marked by multiple different markers, are not being specified properly, right? Although we will see in a later slide, at later time points of regeneration, if you remember, the muscle fiber organization at the tip sort of becomes normal-ish, so to say, right? And so at later time points when that forms, you do see some pole cells organizing, but the initial burst of pole proliferation that is necessary for the early time point head regeneration, no, that's not happening, right? Now, if pole is not regenerated properly, then there is a very high possibility that the patterning molecules, the morphogens, their expression would most likely go bad. So we did an RNA sequencing. We did RNA sequencing at the same time point, 3 days post amputation. And indeed, we found that compared to our controls, the expression of different well-characterized and studied morphogens at this point of time was really aberrant. And we validated that with RNA fish, quite a few. Now, while we were doing these fish screenings, these are predominantly the downregulated ones, one candidate specifically caught our attention, and that was Wnt1. Now, Wnt1 is a canonical Wnt beta-catenin signaling ligand, right? And we saw that at very early time points of regeneration, there was a massive proliferation of Wnt1 positive cells in absence of DDX24, suggesting that at these early time points, DDX24 was most likely repressing the expression or the extent of their expression, right? Now, this was a very encouraging piece of data, and I'll tell you why. Now, the first thing one can ask is, what is the consequence of this elevated canonical Wnt signaling mediated by Wnt1, right? Now, in these flatworms, as is the case with most animals across bilaterians, canonical Wnt signaling is needed for specifying tail fin or posterior identities, and it suppresses head or anterior identities. Therefore, it's not surprising that even in intact planarians, you see a gradient of canonical Wnt signaling with the maximum being at the tail and minimum being at the head. Now, we wondered if this has any physiological relevance, as in like the expansion of Wnt1 in DDX24, does it actually exacerbate the phenotype that we see? Now, to do that, we started off by a very simple experiment. What we did was we started cutting planarians progressively posterior. So there are 3 cuts, AP1, AP2, AP3. AP1 cut is right below the eye, which means that even within an animal, that's the least Wnt environment. AP2 is right above the pharynx, and it has a moderate or intermediate Wnt environment. Whereas AP3 is kept in the posterior region, meaning you have a very high amount of Wnt environment. So now, if you have 3 different cuts with 3 different Wnt environments, low to high, and then in DDX24 RNAi, you have an overproliferation of Wnt1, do these 2 Wnt environments synergize the phenotype and give us a degree of phenotypic severity? And this is what we observe. In AP1 cuts, you still see animals, DDX24 knockdown animals, regenerate rudimentary eyes, whereas in AP3 cut, you almost don't see anything.
Hi, Shordeep. Sorry to interrupt, but you have a few more minutes remaining. Please try to wind up in 2 minutes. Yeah.
Yeah, I'm done almost. Yeah. Please don't worry. I'll finish up. Yeah. Okay. And we went ahead and corroborated this with staining different parts of the eye using opsin and arrestin. And staining was not enough, even though these are pretty images. We wanted to quantify the number of opsin positive, regenerated opsin positive cells. So the first graph shows that regardless of where you cut, the number of opsin positive cells regenerated are less than that of their controls, which is not very surprising, right? But the second part of this analysis is interesting. If you just compare within the control group, regardless of where you cut, the number of opsin positive cells regenerated is almost the same. The same does not, however, hold true for DDX24 knockdown animals. The severity of the phenotype becomes more as you cut more towards the posterior, meaning higher the Wnt signaling, higher is the defect. However, one very interesting thing that we found was that even when you cut in the AP1 plane, which is the low Wnt environment, even though opsin positive cells regenerate, they are heavily mispatterned, consistent with DDX24's role in maintaining muscle, thereby maintaining positional information and patterning cues, right? Now, since there is an elevation of Wnt1, right now, the idea is, okay, can you rescue this defect? So the idea is you knock down Wnt1 with DDX24 and can this defect be rescued, right? And indeed, we see the same, that the number of animals that were able to regenerate photoreceptors after Wnt1 second knockdown was higher compared to DDX24 single knockdowns. And finally, we wondered, like, why exactly does it do? Like, can we have some mechanistic idea about why the second Wnt1 knockdown partially rescues this phenotype? And the hypothesis was that we know that the early Wnt1 proliferation inhibits pole formation. Now, since we have a higher amount of Wnt1, we probably might have a higher amount of inhibition for pole formation. That explains why head formation was defective, right? And this is exactly what we see. That, and I told you in the past that by 8 days, some pole cells come because the muscle fiber organization is sort of rectified in this region by this point of time. But on top of that, if you again lower Wnt1 by knocking it down, you have higher number of pole cell regeneration, and that's exactly how these animals can regenerate photoreceptors better, or regenerate overall better when there is a second knockdown of Wnt1. And this was the working hypothesis that we have, that it works through two parallel ways, one for muscle fiber organization at the tip, and one suppressing Wnt1 at early time points of regeneration. And these two synergize to make the DDX24 knockdown phenotype so severe that we see, that is not otherwise seen in our control animals. This is a published paper, and I'm happy to talk after this, or if you would want, you could see our paper. And yeah, thank you for your time. Thanks to my mentors, my co-authors, lab people for this entire exciting journey. And yeah, thank you for your time. Yeah.
Thank you, Sharadeep, for sharing your insights and showing us all these cool images of planaria. Share the screen. Sharadeep, please stay on for the question and answer session. Thank you. So I hope the slides are visible. So in the next story, we'll hear about a collaborative work between Professor R. Sodamini, Swati Kaushik, and the collaborator Catherine Etchebest lab from InStem in Paris. So this is about prolyloligopeptidases. So we had heard about prolyloligopeptidases or POPs in the sequence theme when we had looked at the sequence search approaches in different bacterial genomes. And so we had Swati talking about the same, and then we also had Soumya, who spoke about the therapeutic potential of prolyloligopeptidases in celiac disease. So now we have Soumya, who will be who's here with us. So she'll be talking about the normal mode analysis of the prolyloligopeptidase protein. So she'll talk about the differences between the ligand bound form and the unbound form, and the insights gained from the normal mode analysis of POP. Over to you, Soumya.
Thank you, Meenakshi and ma'am. So
I hope you can see my screen. Coming to the structure of proline oligopeptidase, this is the structure of a proline oligopeptidase. So henceforward I'll call it as a POP. So it has two domain architecture. So this is the end of hydrolase, which is a discontinuous domain, and then you can see a beta propeller domain in purple, and then the C terminal side of the alpha beta hydrolase. We can see it clearly this side. So alpha beta hydrolase similar to other alpha beta hydrolase, but the beta propeller has a unique open Dasaradhi topology where 7 propeller blades are arranged in a radial across a central tunnel, and the 7th and 1st blades are not connected to each other. So there are several crystal structure available for bacterial POP where we can see the open and closed form. In the open form, the both domain are apart from each other. So there is a channel for substrate entry, but no such open form were found for the eukaryotic POP. Hence, we have studied the porcine POP using both the ligand bound and unbound form, and using normal mode analysis and MD simulation, we can propose the mechanism for substrate entry. So there are various possible mechanism of substrate entry which are already proposed. So first one is as we can see, because of the propeller unique topology of the propeller, there is a propeller pore. So it has been suggested that propeller pore can increase, leading to the entry of the substrate. It has also been suggested that because the 1st and 7th blade of the propeller are not connected to each other, so they can come apart, and it can create a channel for the substrate entry. Also, interdomain opening could be found, so it could also present a channel for the substrate entry. Also, there is a loop called I loop, interdomain loop. The movement of this loop can also facilitate substrate entry. So these are the various possible mechanism of substrate entry which is already proposed. And so we have used normal mode analysis. Because by doing MD simulation, we cannot very easily study the large scale movement of the protein. Hence, we have used normal mode analysis. This is based on the approximation of potential energy. We can get eigenvector and values, and those eigenvectors can represent the natural motion of the protein. Here, the protein is represented as elastic network model, where each atom is represented as beads, and it is connected by other using the strings. And we have used the 100 lowest frequency modes, and those are the most biologically relevant modes, which has very low frequency. So from the normal mode analysis, we have observed various different motion of the porcine POP. So these are the various mode, like the P mode and domain opening mode. Here in the P mode is the propeller of pore mode. Here we can see that these residues are in the interface of the pore, and they usually limit the diameter of the propeller pore. But during our analysis, we have seen that the distance of these residues from the pore increases, suggesting that the dimension of the pore increase. This is one of the motion, and we have also seen that this propeller opening could be is because of the motion of the blade of the propeller away from the tunnel, and not because of the local loops fluctuation. And this is also tied up to the up and down movement of the N terminal alpha beta hydrolase, which could also create the opening of the two domains. So the various types of motion we found in the found during the normal mode analysis is the first one is the breathing in and out of the protein. So when large scale motion of the protein like subunits or domain, when the motion of those are involved, it is called the breathing in and out of the protein. So here we can see the both beta propeller and the alpha beta hydrolase simultaneously moving, and so this is also we call the B mode. And also the twisting motion of the protein is found that both of the domain twisted in opposite direction. So other motion observed in the eukaryotic pop, that is porcine pop, is T mode, which is the twisting mode. Here the two domain twisted in opposite direction, where beta propeller in the clockwise direction and the alpha beta hydrolase in the anti-clockwise direction. So both the N terminal alpha beta hydrolase and the C terminal alpha beta hydrolase, the simultaneously they move together. Similarly, the propeller, all the blades also move together. So it is also observed that so both the domains are required for the movement for this T mode, because if we consider the normal mode analysis of individual domain, we couldn't see this two T mode. And also this alpha beta N alpha beta hydrolase up and down motion, also this induces this motion, because when in silico we delete this N alpha beta hydrolases, no twist motion is seen. And also the other type of motion found is the interdomain opening, where both the domain open in opposite direction, and this is further confirmed by the experimental validation, because cross-linking the propeller and peptidase, it also shown that the domain the enzyme activity is reduced. So we have used the normal mode analysis to see the large scale motion, but the small scale motion, like the movement of loops and kinks in the backbone, so those kind of motion is difficult to study using the normal mode analysis. So for those kind of motion, we have used MD simulation. So here we can see the increase in the beta propeller pore dimension here. So this is the 2 MD simulation in 0 nanosecond and 100 nanosecond. And the magenta are the residues which line the pore. After 100 nanosecond, we can clearly see the propeller size increased. But as we have seen the further experiment from docking and all, we have seen that this size is not enough for a bulky molecule like the T amino acid peptides going through it. So it is possible that small molecule, small inhibitor molecule could go through this, but not 30 amino acid molecule. And here we see here that distance between these residues are found to be increased from 1.5 to 4.6 angstrom. So taking into account all of the motion we found from the normal mode analysis and MD simulation, we can propose a possible mechanism for substrate entry in bacterial pop. So here we see that the unbound form of pop is a closed form, where both the domain are together. And the substrate cannot enter here, but when the up and down movement of alpha beta hydrolase N alpha beta hydrolase lattice, this induces a twisting motion of the two domain here, where the two domain twisted in opposite direction, and this induces the intermediate stage. And in the intermediate stage also, the loop, the intermediate loop for line loop, it also goes from conformational changes. This also involves the loosening of the interaction between two domains, so that they can further come apart and they can create a channel. So in the tunnel, this substrate now can enter and go to the active site. After the substrate bound to the active site, this whole molecule achieve a closed state, and then this is closed state. So this is the mechanism. So we propose after so getting from the normal mode analysis and the MD simulation. So here, thank you all. So please post your question if you have any.
Thank you, Soumya. So we beautifully explained about the mechanism of action of eukaryotic and bacterial prolyloligopeptidases. So I'll introduce the next topic. So for the next topic, we have like use of studies such as virtual screening and molecular dynamics studies to understand the function of proteins. So the third speaker for today is Vikas Tiwari. So he'll be talking about insect odorant receptor. So we have discussed about insect ORs before. So in the structured theme, we had discussed about the insect OR web server, which can be used for identifying odorant receptor gene sequences from insect genomes. And we had also discussed about the molecular modeling of insect odorant receptor and co-receptor in the structure. So today we'll move on to the function theme where Vikas will talk to us about the use of virtual screening and MD simulations to identify repellents for mosquito OR. Over to you, Vikas.
Thank you, Meenakshi. I hope my slides are visible. Good afternoon, everyone. And in this talk, I will be discussing about identification of potential repellent molecules against the mosquito Aedes aegypti by targeting a specific potent receptor through virtual screening approaches. So generally, all the insects rely on olfaction for performing various activities like predation, foraging, or kin identification. And they rely on several classes of families to perform this olfactory behavior, which includes odorant receptors, gustatory receptors, and ionotropic receptors. And in this study, we have focused on odorant receptors, which are comprised of odor-specific protein called odorant receptor and its co-receptor, which is required for functioning of all the ORs. And there are no experimental structure available for any of these ORs. And especially in cases of disease vector like mosquito, it has been shown that a particular OR called OR4 is responsible to impart this mosquito human host preference. Therefore, this OR4 and its co-receptor becomes a natural target for designing repellent molecules. So as there are no structural information available, we started by employing computational structural modeling techniques to first get the 3D structure of the co-receptor and OR. For this, we used the template structure from Pig-Wasp co-receptor, which is a homotetramer. And we took one of the monomeric unit as a template, and by applying homology modeling techniques, which I had explained in the structural theme of this CAPS talk series, we had the final model of the co-receptor, and similar way we had modeled the OR4 structure as well. And once we have this static structure, we subjected it to a short MD simulation run in membrane environment to get a final structure at the end of the simulation, which had no residue in the disallowed region of Ramachandran plot. Further, we could also identify some of the conserved residues, like a triad of polar residues in the transmembrane helix, and also the residues at the end of the pore-forming helix. Once we have this final structure, which is energy minimized in native condition like in membrane environment, we could identify the potential drug binding sites on the surface of this particular protein. And we can go ahead with the virtual screening protocols. So this protocol generally involves the availability of the 3D structure of the protein, which in this case can be ORCO or the OR4. And then we can identify druggable sites on this particular protein. And then we generate a grid around these identified sites. And for ligand databases, we have used the known repellent molecules like DEET, along with several natural products from this database called Supernatural 2 database. And then we prepare these ligand molecules by generating various stereoisomers, adding hydrogen atoms, and so on. Once we have the protein and the ligand, we can go ahead for the virtual screening, which involves several steps, which generally includes high throughput virtual screening, standard precision docking, and extra precision docking in the order of increasing stringencies. And finally, we have a set of potential binding ligands, which can further be assessed through delta G calculation or the molecular dynamics simulation. Just to give one against one side the results. For high throughput virtual screening, we could identify around 2 lakh possible ligands. And when we compare the energy of the top scoring ligand with that of the positive control, so at each step, we have a positive control compound, which has been verified experimentally. So we can see that the positive control compound had a less or worse energy compared to the top compound. So we take some top 10% of this compound and we proceed for the next step. And each step we have this positive control compound to compare with. And similarly, we perform the last step, which is the extra precision docking. And finally, for these 2000 odd ligands, we can calculate the delta G scores and again re-rank these molecules and pick top 10 hits and assess their interaction stability through molecular dynamics simulation. So here I will be showing one of the kind of analysis for one of the ligand bound to co-receptor, which is ortho. So here in the beginning of the simulation, you can see that it is involved in multiple interaction with several residues. And once we subject it to simulation, we found that the ligand remains stable and remains bound to the protein as assessed by the root mean square deviation plots. And we can also see like which atom of the ligand is interacting with which residue. So we can see some of the residues which are there in the beginning of the interaction. They remain interacting throughout the simulation time with the ligand. We can also assess the different kind of interactions through which the ligand is interacting to the protein. And we found that some of the hydrogen bonds, they persist for more than 80% of the simulation time, indicating that the ligand remains in its binding pocket throughout the simulation time. And also, we assess the total number of contacts across the simulation time. And at each time point, there is at least one interaction of the ligand with the protein. So similar procedure was carried out for the OR4, which is responsible for human host preference. And in this case also, we started by structural modeling and then doing short MD simulation. And finally, we could identify a potential drug binding site, which is on the extracellular site of this protein. And in this case also, by following several rounds of docking protocol and MD simulation, we could identify several hits. And here I'm showing one example data for one of the top hits. So in this case also, we can see that the interaction it maintains in the beginning of the simulation. So for example, with the glutamate 200, and it keeps interacting throughout the simulation time as assessed by the persistence of these interaction plots. And also in this case also, we could find that all the ligand remains in bound form with the OR4. To summarize the top hits for these two different receptors, we could identify several ligands with a common name, and also we can trace back their source plants, which include some interesting hits, like some of the compounds we could identify were from black pepper, from tulsi, from red pepper, and also from white mulberry. And some of these plants, like the tulsi and the white mulberry, has been shown to have insect repellent activities. Further, all of these compounds have better binding energy than the positive control compound or the known repellents. So therefore, they are very good candidates for repellent development. So further in vitro experiments like electrophysiology or the behavioral assays can be carried out with these compounds to establish these as a potential repellent molecules. Thank you, and over to you, Meenakshi.
Explained very well about the methodology used for virtual screening and the MD simulation and post-MD simulation analysis. So now we move on to the last talk for today, which is, I'll just share the screen. So we have with us Abhishek Sharma. So during the initial few months of the SARS-CoV-2 pandemic, so a lot of labs throughout the world had been looking at effective strategies and ways to inhibit the SARS-CoV-2 virus. So our lab also joined in these efforts, and we looked at one of the proteins, which is non-structural protein 1, the NSP1 of the SARS-CoV-2 protein, and we looked at the structural modeling and virtual screening studies for the inhibition of the SARS-CoV-2 protein. So Abhishek will talk about the same. So over to you, Abhishek.
Thank you, Max. Hi, everyone. Myself, Abhishek Sharma. I'm a joint student between Professor Sudhamni and Professor Sudhir Krishna. Today, I will talk about our effort in the COVID era for screening approximately 3 lakh small inhibitors belonging to FDA-approved as well as plant origin against a COVID-19 protein called NSP. So this presentation is divided into 4 parts: objective, methodology, which we followed, and finally results and conclusion. Our aim of this study was to screen these small inhibitors against a potentially small proteins called COVID-19 NSP1 protein. SARS genome is around 30 kb long, and this is divided into 4 genera: alpha coronavirus, beta coronavirus, gamma, and delta. The viruses with alpha and beta coronaviruses contain only NSP1, and it is only a small proportion out of the whole genome. This protein, only found in 2 genera, its sequence is highly divergent as compared to the within the coronaviruses, and the length of NSP1 is also varied from 180 amino acids to 250 amino acids. So now the question is why, when all the world are chasing against spike protein, we are looking for an NSP1 protein. And the answer to this is the role of this protein in the disease pathogenesis. So here in this chart, you can see that the role of NSP1 in other coronaviruses, where it is inhibiting the translation, inducing the host RNA cleavages, and so on and so forth. Therefore, we choose this protein because it acts as a virulence factor, by inhibiting post-translation and subsequently inducing post-mRNA degradation. This favors viral protein expression. Also, it makes host more susceptible by chemokine dysregulation by inhibiting type 1 interferon signal. Now, what is the recipe of the viral virtual screening against NSP1 protein? When we start this project, there were no well-defined structure of NSP1 in the PDB database. Hence, we go for the homology modeling, where we take the solid structure of SARS1 NSP protein 2HXX as a template, and then perform the homology model. After evaluating the structure, we go for the main part of the virtual screening, that is identification of a druggable site. Ideally, a druggable site should have two components. One, it consists some kind of residues which are critical for the protein functioning, as well as should we have some accessible sites where the small inhibitors can interact with those residues. So for the first part, we did the sequence alignments and identify some such kind of residues in the Wuhan NSP1. Important residues which are shown to play a role in affecting host gene expression and antiviral signaling are highlighted in the green and purple color in this alignment. We use Schrodinger's tool called sitemap for predicting the sites which can accommodate small inhibitors. There are two sites which are predicted by us. One is the which you can see in the blue called deep site, and the other site is shown in the red called shallow site. Now after this, we follow the virtual screening pipeline. For this, we take around 3 lakh compounds from two databases, Supernatural database and FDA-approved database. The steps of the virtual screening are explained by Vikas in the previous steps. Hence, we firstly prepare the ligands via LigPrep and pass these ligands to the virtual screening pipeline. At every step, in this case, we take the top 10% of the compounds and pass it through the next step from STVS to SP to XP step. Finally, the compounds which obtained from the XP extreme precision step were further refined by MMGVSA algorithm. The compounds which refined by the MMGVSA algorithm were passed to the MD simulation. The MD simulations were carried out to check whether these compounds indeed remain interact with the protein for longer duration of the time and are not just an artifact of the virtual screening pipeline. The relationship of the MD simulation and using virtual screening pipelines is well explained by the Vikas in the previous talk. So I will directly jump to the results. By this pipeline, a set of potential inhibitors were obtained, which include FDA-approved antiviral drugs as well as anti-inflammatory drugs. We also identify a set of small inhibitors from the plant origin, which already have shown to have antiviral, anti-inflammatory activities. More interestingly, this study shows the role of publicly available datasets in the fast drug discovery. So our study starts with a literature survey where we have no information about this NSP1 protein. We made a model, validate it, followed by we prepare the small molecules and protein for the virtual screening and carry out the whole step for the virtual screening pipeline. This whole step were approximately accomplished in the 17 days. Finally, from this study, we identify a set of potentially FDA-approved and plant origin small molecules. Some of them are in further validation within the collaboration. And this approach showed an important role of publicly available data in the identification of potential drugs in short period of time. Thank you. Over to you, Akshay.
Thank you, Abhishek. So this was very insightful. Thank you for sharing the strategy, like why the NSP protein was chosen and followed by the methodology virtual screening and the results. So I will share the screen for a summary. So today we discussed 4 talks. So we started with Dasaradhi's talk. He spoke about the role of DDX24 in the planaria bone regeneration and what defects arise when DDX4 knockout situation is created. Then we heard from Soumya about Swati and ma'am's work on the mechanism of action using normal mode analysis between the ligand bound and the unbound form of eukaryotic and bacterial prolyloligopeptidases. Then we again heard from Vikas about the virtual screening strategy used to identify repellent molecules for insect, that is mosquito odorant receptor. Then at the end, we heard from Abhishek about the SARS-CoV-2 NSP1 protein, but why this particular protein was chosen for the analysis, and then the drug screening efforts, screening for the naturally occurring molecules followed by the drug-like molecules. So thank you to all the speakers, and say if there are any questions, we can take the questions. So I would like to request Shafi to share the references for today's 4 talks.
Hello, everyone. So these are the relevant publications related to today's talk. So the first publication, Saldik talked about DDX24, and following to that, Soumya talked about Swati Kaushik's work about prolyloligopeptidases. Then Vikas talked about organ receptor modeling, and then following to that, at the end, Abhishek talked about COVID-NS1. Yeah, so this is the end of actually the last talk of function theme. Now, from next week onwards, we will be moving to the next theme, that is medicinal plants and collaborations. And first talk will be on computational studies of plant stress management. Please stay tuned.
Thank you, Shafi. So now so since today is the last talk in the function theme, so I'll share some consolidation slides. So in the first day, so we had R-struct FAM being discussed, that was by Preetha. Then we also discussed the story on TGF receptor by Parantu and Christina. Then we had a discussion on the YLED protein by Harini, followed by deaminase and deacetylase proteins by Vikas. And on the second day of the function theme, we discussed about the Hedgehog protein. So coincidentally on these days, all the work that we discussed were collaborative in nature. So the Hedgehog protein was also a collaborative effort. Then even with the generalist OR work discussed by Shrishti, this was also a collaborative work with Dr. Shannon Olson's lab at NCBS. And the Hedgehog protein, so this protein was also a collaborative effort with the labs of former and the current director of NCBS. Then we discussed about the tropomyosin work by Margaret Sunita. So this again was a collaborative effort with the James Goodrich lab and InStem at the Bliss campus. Then we also discussed about MTM1 protein, where we had collaborated with Dr. Abu Padinja's lab. And about the PAZ proteins, where we worked, Nitish had spoken about this work, and we had worked with Dr. Ramaswamy lab and Dr. Vinod Kumar lab. And the MTM1 work was explained by Tina. So on the third day of the function theme, so we had CFG, conserved functional groups explained by Axel. Then we had FASSM, so ma'am spoke about this work by Gaurav. Then we had the cascade approach. So Sandhya had presented this work. Then we spoke about the CAPS protocols with an application of functional annotation to transcriptomes, plant transcriptomes. So this talk was given by Abhay. Then, so today, so we have Sauradeep. So currently he's a postdoctoral fellow at Stanford, and he was a joint PhD student with Sodamini ma'am and lab at NCBS and Dr. Dasaradhi lab at InStem. And we have the normal mode analysis of prolyloligopeptidases work, which was done by Swati and explained by Soumya. Then the virtual screening and MD simulation work for SARS-CoV-2 work was explained by Abhishek. Then the virtual screening for organ receptor of the mosquito Aedes aegypti, this work was explained by Vikas. So currently Soumya, Abhishek and Vikas are at Sadamini lab, are current students of the lab. So thank you everyone, and please stay tuned for the CAPS talk next week. Same day, same time. And thank you for joining us. Keep supporting us the same way next week. So we meet every Thursdays at 3 p.m. for the CAPS talk series.